potassium pump, a neuron would be able to create electrical potentials rather, and as a result wouldn't be able to transfer information really, really quickly. So a long time ago I was in grad school and I was sitting in a couch and I looked like a ninja, right? And I'm eating something, someone goes, Mike, and I turn around like this and I go like this, and I caught an orange, like right here, and I looked like a ninja. I was like, everyone was like, holy crap, and I was like, how the hell did I do that? And then I said, why are you throwing oranges at me? And I thought you were looking, I was just throwing your arms up. Why do you do that? Like, go on. So how did I react that quickly? I have no idea. Okay?
You know, like your Twitch fibers?
Well, so at the cellular level, there was the sodium-potassium pump that created this membrane differential. So there was a bunch of potassium on the outside, a bunch of potassium on the inside, a bunch of sodium on the outside, and these things, and I'll see if you remember what they're called, integral proteins or channels that opened up very, very specifically, and I was able to go like this and look like a ninja. So when you're talking about the sodium-potassium pump, think of me touching things like that. I looked awesome doing it, by the way. And everyone was like, oh, did you see that? So how does that happen? The only way it happens is you have to get set up. You have to get it set up before that trigger. That has to get triggered. Because it's no value if you need to set it up while this thing is coming in the air, right? You got to be ready to respond immediately. The way that you respond immediately is through this ATP pump. And that ATP what happens here is that if this is in the cell, and you guys are all potassium ions, I'm going to ship 80, 90% of you out of the cell, okay? Through that channel right there. I'm going to open up the door, I'm going to ship you all out. And, sodium channels. And then when the time is right, I'm going to open the door and you're going to rush right in. So that membrane, it's called a membrane potential here. And not on here, but the membrane potential, the difference between the concentration of an ion between one side of the gradient or one side of the phospholipid bilayer and the other is required to get set up. Okay. So the example here is a sodium-potassium pump. And this happens throughout the body, and it's very important, and we're going to talk about it throughout the semester, including at the end of the semester when we get to the neurons and the nervous tissue. So that sodium-potassium pump is an active process right here. So if you look in the upper right-hand corner, I'm just going to talk about this cartoon right here on the right. You can read along on the left. But on the right-hand side, any sodium ions that are found within the cell get to this phospholipid bilayer, this specialized receptor, that integral protein. Right? It grabs those, in this case, I'm not going to worry about numbers until later on in the semester, but it grabs sodium ions into the cell and it ships them out. In the process of shipping them out, while it also grabs potassium ions and it imports them into the cell. The only way that happens here, that's one cycle of the process here, sodium out, potassium in. The only way that happens is through the use of ATP. So that ATP right there is now hydrolyzed or decomposed, right there. And so if you go down at the bottom here, the potassium is shipped into the cell and the sodium is shipped out of the cell. So that ATP is hydrolyzed, and what does that mean, hydrolyzed? You're going from ATP that way. It's broken up. Okay? So once again, the concentration gradient between the sodium and potassium is very, very high. Lots of sodium outside of the cell and a ton of potassium inside of the cell. Then when the time is right, when you got to be a ninja, those receptors open up. There actually turned out to be different receptors, but there's receptors, and they allow sodium to rush in and the potassium to rush out. And when this whole process occurs, this will occur on a scale of 1,000th of a second. So they're called milliseconds. maybe 2 milliseconds, 3 milliseconds is how quickly this happens. None of that will responding to stimuli, you can't happen there unless you have it all set up, and this is the way it sets up. Look at this graph right here. Now this graph is not from your A&P class, it's from a nursing book that I got from, I think I got this in 2017 or 18. And it's awesome. But I want you to sort of orient yourself a couple things. First of all, so this is a basic nursing book, and if you look at the top, there's 4 columns. There's 2 on the left, 2 on the right. There's on the 2 left-hand side, they're called the plasma, and the 2 right-hand side is intracellular. So intracellular is something we already used. We haven't talked about what plasma is. Plasma is also called extracellular fluid. Okay, so plasma and extracellular fluid, blood plasma, the liquid nature of portion of blood here. From the nursing perspective, that's all we're talking about here. Anything that's fluid in the body is probably plasma, okay? Unless it's something different. So depending on location, the liquid will change. Each of these columns here, each of these outside of the cell or inside of the cell have 2 ions. There's cations and anions. Cations and anions. I still want to say cations now.
Just say it.
Cations.
Just say it.
So I was thinking about it the other day. I'm still laughing about that because I never even dreamed, I never even looked at it that way. And that question that you asked me, I was like, it totally upset me. Anyway. So now there's cations, there's anions. Are any of these cations or anions familiar here? Well, look at this one, potassium and sodium. Where is the sodium? Now, and then final thing is look at the size of the box. The size of the box means the concentration across this line right here. What's between these 2 columns here? These 2 columns on the left and 2 columns on the right. What's between it? A phospholipid bilayer. Okay? So this means that the sodium concentration is greater outside than inside, and conversely, the potassium concentration is greater inside than outside. How does that happen? The only way that happens is what I just told you, through that ATP ACE, that ATP powered pump, the sodium potassium pump. Gets shipped out of the cell or into the cell, depending on the structure. That also happens other things too. So if you look at chloride here, how much chloride, relatively speaking, is inside the cell? Very little. How much is outside of the cell? An awful lot. I mentioned chloride concentrations and chloride membrane or problems with chloride concentrations earlier on in the day, and that's with cystic fibrosis. And so that if the concentration gradient is not appropriate, then it causes the body to respond in a problematic fashion. So in this case now, we have, whenever there's a big square or rectangle on one side and a little one on the other side, that means there's a concentration gradient. Where is there not a concentration gradient? Look at this thing. Right? So now there's magnesium. Magnesium is a very important element or ion. Right? What happens if the cell membrane, which would be right here, what happens if the cell membrane opens up to all magnesium concentrations? Not much. Why? Because it will pass through a little bit, and then some will pass. There's not a hell of a lot that's going to happen. What's going to happen if it opens to potassium? And this will be a question tomorrow. Here's the graph. What happens if the membrane allows for the transport of potassium ions across the phospholipid bilayer? In which direction would it go? It goes from intracellular to extracellular, or from intracellular to the plasma.
Will we have access to
You would be given this,
Okay.
right? So you will be given this graph, and I'm going to ask you to interpret it. It doesn't matter if you're talking about a cation or a cation or an anion, but what you have to do is you have to remember that this is the phospholipid bilayer right here, and in both cases you have special transporters or integral proteins that allow for the transport of these ions from one side to the other. In all cases here, all of these are positively charged, or not positive, they're all charged. The other thing is, you see this thing right here? That's a protein. How do you make a protein? You know this. You assemble amino acids through dehydration synthesis. You string them along a lot together. If the R group here, if the side chain of the amino acids have lots of negative charges on it, then you get a negatively charged protein. If you got a lot of positively charged R groups on that amino acid, then you get a positively charged protein. But for this case, right, for our cells, we're worried about anything that, in this case, negatively charged proteins. Okay, so how is this created? Right, and when I say this, the sodium-potassium concentration gradient for the sodium-potassium pump. What causes that, or what allows for that for ATP? What type of transport is that? That's active transport. If the cell membrane opens up channels and allows for the passive movement, in which direction will sodium move? Sodium will move into the cell. In which direction will potassium move? Potassium will move out of the cell. How do I know? It's based on the concentration gradients. Okay? And once again, just for sodium potassium, the only way that's set up is through the active transport. Okay? Questions? So this sodium-potassium pump, you have sodium getting pumped out of the cell and potassium getting pumped into the cell. This question right here, this pump right here, what would I call this in the context of this class right here or this lecture? This could be an integral protein, it could be a receptor. It's an example of active transport right here. When you're taking ATP and you're making ADP, right, that's got to be active transport. Active transport, generally speaking here, requires ATP and it moves it from a concentration gradient from a low concentration to high concentration. Now, what happens if you look on the right-hand side and you have a certain amount of glucose in the cell, and you need to dump more glucose in the cell, and more glucose in the cell, and more glucose in the cell? What happens to your body? Is that good for your body? No, right? That's a problem. So that would be something like, ultimately, if you get too much glucose into your pancreas, that could be diabetes. Right? So that's there. I'm not going to ask the distinction between primary and secondary active transport. We're just going to talk about active transport. A vesicle. If this is a cell with a little bit of these sort of, if you guys know what Pac-Man is, right? That used to be one of the only video games that were around in the world not too long ago. But if you look inside the cell intracellularly, this is called a vesicle. A vesicle is anything inside a cell that's surrounded by a membrane. And what kind of membrane? A phospholipid bilayer. That vesicle, hold on a second, I got an ice cube. That vesicle, I did not want that. That vesicle could either be coming into the cell or it could be in the process of getting out of the cell. If it's in the process of getting out of the cell, so now we have to change this picture right here. We'd have to make it, then that would not be this part right here, it would be this part right here. So that if the cell made this thing, let's just say these are enzymes that's going to digest something, then it would try to get out of the cell. And that's called exocytosis. So generally speaking here, there's endocytosis and there's exocytosis. And there's two types of endocytosis. When you have small particles getting ingested, it's called pinocytosis. And when you get large particles, it's called phagocytosis. An example of phagocytosis, if you have an infection. So my wife had COVID a couple weeks ago. By the way, I keep saying this. I don't know why I haven't gotten it. Staying away from all you guys.
You haven't had it yet?
Well, I had it like 2 years ago. Right? Or
That's impressive.
it might be 3 years ago, actually. It's been a long time ago. I mean, I'm getting my vaccines and stuff. I'm the first person in line for a vaccine, just in case you're wondering. I'm absolutely. but I can't figure out why, my daughter had it, my wife had it this fall. It was like 400 people at Rana have had it. Not 400. A lot of people at Rana had it. I don't know why the hell I don't have it, but anyway, I digress.
Don't jinx yourself.
What?
No, for real, don't jinx yourself. Not
I know,
gonna-
too late.
I've had it twice, even after I got my vaccines. It was,
You're lucky.
yeah, so lucky. My brother's had it like 3 times, so.
So
That's like-
phagocytosis, what happens in phagocytosis? Your body gets invaded by bacteria or viruses. COVID, the COVID virus, for example. It gets recognized by leukocytes. And what's a leukocyte?
White blood cell.
A white blood cell. It gets recognized by a leukocyte as an invaded particle. Right? It phagocytizes or engulfs that, and it breaks it down and digests it. So phagocytosis and pinocytosis are 2 examples of endocytosis. Exocytosis is the release of something. And I'm not going to talk about transcytosis or ask about it. So pinocytosis illustrated here on the left. Another example of endocytosis I mentioned earlier is receptor-mediated endocytosis. What is that? So a receptor is something that is attached to the surface of the cell that recognizes particular particles. Those particles then get engulfed or into a vesicle. Right here, and if you look at the cartoon here at the top, there's several things that are attaching to those receptors, those several chemicals here. As you go down to the middle panel here, there's more and more, and slowly but surely the cell membrane then engulfs upon itself and it imports something into the cell. That's also an example of probably pinocytosis, small things. Phagocytosis, we're going to just say it's invading particles, particularly dead material, dead cells or broken cells, bacteria and viruses. So that's an example for phagocytosis. So if there's anything else but phagocytosis, it's pinocytosis. So here's an example of pinocytosis, but the receptors are helping bind it. When you guys eat something, it goes into your stomach, and your stomach sends out hydrochloric acid. That hydrochloric acid could burn your body. If you took that hydrochloric acid and it was just the hydrochloric acid, you put it on your skin, it would hurt. It literally would burn your skin away. If you have too much hydrochloric acid in your stomach, you get ulcers. It literally eats the lining of your stomach.
Aren't we protected by like the mucus in our stomach?
So the reason why that doesn't happen under normal conditions is because we're protected by a number of things. One of which is mucus produced by the stomach, that lining. And then the other thing is that if you have an appropriate amount of hydrochloric acid, you hit the food with hydrochloric acid, the hydrochloric acid gets used, and then it gets passed out of the stomach, and so then that's another reason, right? The cells that make hydrochloric acid, if they don't want to digest themselves, right? You want to be making it and then get it the hell out. How do you get it the hell out? Through exocytosis. If you look at this, here is a cell with these bubbles right here, and imagine these are hydrochloric acid. If this thing breaks, everything around is going to get decomposed. Right? You don't want it to break. The cell doesn't want it to break. So what happens here? It ships it out to the cell, and then it gets released. So when that vesicle is expelled from the cell into this extracellular fluid, right, then it encounters another environment. So endocytosis, exocytosis. So now we'll talk a little bit about organelles, and I've already talked about this stuff. So this stuff should be very familiar. We've talked about the cytosol or the cytoplasm. The liquid portion of the cytoplasm is the cytosol. I haven't mentioned much about the cytoskeleton. So the cytoskeleton is, I talked a little bit at the drawing at the start of this lecture here. The cytoskeleton is basically a series of the internal structure, just like we have a skeleton in our body, and you're going to be tested this week. The cytoskeleton has a couple of different things to it. there are these tubules, and anything with micro in front of it, there's little tubules here, there's little filaments or microfilaments right here, and then there's also filaments that are sort of in between there. There's moderately sized. These in particular, an active actin filament. Actin is a protein that when we get to the muscular system, we're going to talk about its cousin myosin here and doing muscle contractions here. Actin allows for the movement or the shape of the cell. We're not talking about cell contractions here or muscle contractions. We're talking about taking the cell and moving it from this big to this big, or moving it from this big to this big, right? That allows, so we're talking about moving or changing the shape in a particular way. So that actin filaments will help there. Another thing, and we talked about cytoplasmic inclusions. An inclusion is another way of saying a vesicle. So one of the things about this class and about biology and sciences in general is that the vocabulary can be substantial. So I have talked about a number of different ways of organizing or talking about integral proteins, starting from the basic integral protein to a whole bunch of other stuff. And now here's an example of a vesicle is also a cytoplasmic inclusion. Right? Those are the same thing. Generally, we talk about organelles as being membrane bound. Again, those membranes are the phospholipid bilayers. So let's just sort of run through some of these. The nucleus is by far and away the most important one. It sets up the structure of the cell, or the function of the cell rather. It has a couple of different types of nucleic acids, either permanently residing or temporarily residing in there. RNA is temporarily resided, built inside the nucleus. DNA is found within the nucleus. The nucleus itself has a nuclear envelope. So that phospholipid bilayer no longer is the phospholipid bilayer, now it's called a nuclear envelope because of the location. It is the same structure, it's just more strict about what lets in and what it lets out. It lets in the building blocks of nucleic acids. So now I do this all the time, but here's an example of a question. I ask him to start writing a sentence or two about the nucleus here, allowing for the building block and building or having stuff present in the nucleus, particularly RNA and DNA. What's the monomer of a nucleic acid? In that case, right, I'm trying to mix you up. I'm talking about the nucleus, I'm talking about cells. The monomer of a nucleic acid is chapter 2. Right? The monomer of a nucleic acid is a nucleotide. So the nuclear envelope, where my pointer is, allows for nucleotides to pass in, right? It then has a series of enzymes that assembles the nucleotides in a particular way. It either makes DNA or RNA, depending on when we're talking about that. And when it makes RNA, then it ships it right the hell out. It does not allow RNA into the nucleus.
You didn't give me enough time to give
Is
you
what? Oh,
I said you didn't give me enough time to get.
you were going to answer that?
Yeah.
All right, I'm sorry. I'll give you more time the next time. So here's something important, right? The nuclear envelope is very strict. It doesn't let a lot of stuff in. Why is that important? So you hear people saying COVID vaccines will change my DNA? No, right? People have argued that I'm not getting a COVID vaccine because it changes my DNA, and that can't be more false. Okay, here's another thing. I'm a 6'10" basketball player, black basketball player. Right? Aside from being human, none of that is accurate, right? I don't know. I don't think that I'm a 6'10". I don't think I'm black, and I'm certainly not a good basketball player. Right? So I'm 0 for 3 in that. I am human. Right? So is any of that accurate? No. That's about as accurate as saying the COVID vaccine will change your DNA. No vaccine will change your DNA. Your cells won't let it. Your cells don't want it. What happens if your cells allow for the change of DNA? What's that? Well, it's bad, but what's that called? There's a particular disease, class of diseases, when your cells have changes in DNA, it fights like hell to keep it from happening. Your body's not perfect. It's called cancer. All right, so that's what we're talking about here. So if you're talking about changing DNA, that's a problem. Okay. So whether it's DNA or RNA, the DNA is resident in the cell, in the nucleoplasm or the cytoplasm of the nucleus. Right? The RNA is shipped out to undergo what it normally does is protein synthesis and other things. When you're talking about protein synthesis, you're talking about ribosomes or rRNA. If you look at this, there's 3 different types of RNA. There's transfer RNA, messenger RNA, and rRNA, or ribosomal RNA. There are free or attached ribosomes. Free ribosomes reside in the cytosol itself. There's little dots, as in my drawing. Attached ribosomes are attached to an endoplasmic reticulum, or ER, and that makes the rough ER. Regardless of the RNA that we're talking about, it's made within the nucleolus, right here, and then it's shipped out. Talked about RNA being attached to something. If it's attached to an ER or endoplasmic reticulum, it's rough. If it's not attached, it's smooth. Generally speaking, the rough ER produce proteins. The smooth ER produces lipids and carbohydrates, but definitely lipids. Rough ER, let's see here. And then this part right here is a smooth ER. So generally, they're adjacent to one another or near one another. They're attached to the nucleus, so that the RNA will be exported. The rRNA will be exported from the nucleus and immediately attached to the endoplasmic reticulum. So that network is really important. That network is hugely important for the cell, and that network then is attached to this thing right here called the Golgi apparatus. That ER is attached to the Golgi apparatus. I mentioned a little while ago about cells producing hydrochloric acid. Once it produces hydrochloric acid through, let's just say it's at the rough ER, it produces hydrochloric acid, you immediately got to box it up and ship it the heck out. How do you box it up and ship it out? Through the Golgi apparatus, or sometimes called the Golgi bodies. Those Golgi bodies produce phospholipid bilayers, right? And it engulfs something, it surrounds something, and then the process of it surrounding it, once it's done, what is it called then? It's called a vesicle. And what's the process when it gets out of the cell called? That's called exocytosis. A transport vesicle, as I have at the bottom here, is just a substance that's been packaged, built inside a cell, surrounded by the Golgi body or Golgi apparatus, and then exported from the cell. You can read about the functions of the Golgi apparatus, but I pretty much told you what they are. Here's a telegraph. Here's a hint for me. If you wanted to get into this, and sort of, especially at the bottom right here, sort of look at how detailed it can be, you can, but the likelihood that I get really, really in the weeds with the Golgi apparatus is very, very low. Vesicles generally can be come in 2 different categories here: lysosomes or peroxisomes. These intracellular organizations there, they can be formed at the Golgi apparatus, right, or the Golgi body. They may not be. They may be formed elsewhere, and we're not worried about where they're going to be formed. But generally speaking here, lysosomes and peroxisomes for this class are just vesicles. They might have a variety of functions, including digesting parts of the cell, breaking down fatty acids or other things, keeping things that are toxic away from the rest of the cell. So lysosomes and peroxisomes have very similar functions. If you get into cell biology, they're different, but that's not what we're going to do. And this again just illustrates some vesicles here on the right-hand side. And what is this called right here? So that this is the lysosome, it's joined with this vesicle right here. It's gonna digest this and it's called exocytosis right there. I'm not gonna talk about proteasomes this time. Now, have I ever talked about the mitochondria? Have I ever talked about the structure of the mitochondria, the cristae and the matrix? Have I ever talked about the membrane and the cell metabolism associated with it? All right? So if you go back to your notes from last week, everything that I'm going to talk about right now is all in those notes. But I want you to really, really focus on what I'm not talking about. Okay? Awesome. And by the way, what are we doing here? In the mitochondria, we're going from the left side of this equation to the right. How is that going to happen? Through ATPase, that's the enzyme that's responsible for making ATP. So ATP synthesis occurs in the cell and inside the mitochondria, depending on the steps in the process. When you're talking about the mitochondria in itself, we're talking about the infolding is the cristae of the mitochondrion, and the space within the mitochondrion is called the matrix. Regardless of whether we're talking about the outer membrane of the mitochondrion or the inner membrane of the mitochondrion, it's just a phospholipid bilayer. And those things, depending on where you are in the mitochondria or outside of the mitochondria, you have different steps in the process of cell metabolism. When you're outside of the mitochondrion, you're not doing anything inside of the mitochondrion, right? That makes sense. So that when you have glucose getting imported into the cell, into the cytosol, that immediately enters into glycolysis. Okay? Then, when glycolysis is done, you see this thing called Krebs cycle, or the citric acid cycle? Krebs is just some old dead white guy, probably German, that described it first, and he said, I'm going to call it my name. It should be the Clancy cycle. Right? But it's also the citric acid cycle. So after glycolysis, the output of glycolysis gets imported into the mitochondria. Where does it go? So you guys are sitting on chairs, right? When you came in this room, are you sitting on chairs? No, right? You're in the space of the room. Right? I'm in the space of the room. I'm not sitting down. Rochelle came in and sat down. This is a membrane of the mitochondria. Okay? So if you're not sitting down at the table, you're standing up or sitting on the floor. In either case, you're not at the table. If you're not at the table, the matrix of the mitochondrion is the space. When you talk about the matrix of the mitochondrion, you're talking about the citric acid cycle, and you know what happens in the output of the citric acid cycle. You're producing NADH and FADH2 and a little bit of ATP, but also you produce CO2. What happens to CO2? Does the cell use CO2? No. When you produce CO2, here's another question I can ask. The Krebs cycle, the citric acid cycle produces CO2. What happens to the CO2 and what process describes that? What happens to it? It gets exported from the cell. What processes that? That's called exocytosis or diffusion, right? Getting out of the cell. How is that generated? Through a concentration gradient. That's diffusion, right? So again, I started asking about the mitochondrion, and then now I'm asking about diffusion. In the citric acid cycle, we produced two main things here. There's FADH2 and NADH. And if you go back to your notes, that's what we said. Then I'm going to sit, I'm pushing thin out. I'm sitting right here. Now I'm at the table. If I'm at the table, then I'm in the electron transport chain. Three things get into the electron transport chain besides these. NADH, FADH2, and oxygen. You have a whole bunch of hydrogens and oxygen. You combine those with this thing right there, and you make some water. In the process of making water, you make a bunch of ATP. That all happens inside here. Right? One final thing I'll talk about them, and again, if you want to, you should review your notes about the steps in cell metabolism, right? But there's 3 main steps: glycolysis, the citric acid cycle, and electron transport. And we're going to treat that as a single series of steps. There's only 3 steps that we're talking about. I'm not going to get into the the names of the enzymes or the steps in glycolysis of the citric acid cycle. Okay? Major inputs and outputs of these products and these steps, and then finally ending up with a ton of ATP produced. One final thing about the mitochondria. I thought you were going to ask me something. You guys are all genetic clones. I'm a genetic clone. Do any of you guys, well, first of all, do any of you look like me? Oh, that's good, right? Do any of you look exactly like your parents?
No.
Any, got any twins in here? Not fraternal, but identical twins? Does any of you look exactly like your siblings? No, right? Unless you're genetically identical. And yet, and so that if you're genetically identical, that's called a clone, and yet you're clones of your mothers. I'm a clone of my mother, which is awesome. She was fabulous. Why am I a clone of my mother? Because during sexual reproduction, the sperm fertilized the egg, the sperm brought half of the DNA, and that's it. The ovum, or the egg, carries all the cytosolic elements here, everything inside of the cell that we've talked about today. The female brings, the mom brings, one of which is this. And this has a little bit of DNA in it to organize, and that is found just in the mitochondria. That mitochondria, then the DNA directs the reproduction of the mitochondria over and over and over again. And there's no other genetic recombination of that. There's no other changes in the genome. So my mother and I are genetically identical in our mtDNA. Me and my grandmother are genetically identical. Me and my great-grandmother are genetically identical. But me and my father have no nothing in genetic in common in our mtDNA. Here's something really sad. Me and my kids have nothing in common in my mtDNA. Right?
At what point, like for example, for example, your great-great-great-great-grandmother still has the same as you, or did it break off at one time?
It never breaks off. So you can trace your history back thousands of years. There's going to be some changes in the DNA, but you can go back thousands of years and say that you're a genetic clone of those people from 1,000 years ago. Right? It would be a little difficult, but in theory.
How would you figure out, like, you know how there are situations where the child doesn't know who the father is and trying to figure it out? How would you figure that out if you don't have any genetic DNA?
So, wait a minute, I didn't say genetic DNA, all right? I said mitochondrial DNA. Now, when you're talking about genetic DNA, right, nuclear DNA it's called, right? Half comes from dad, half comes from mom. So my kids have half my DNA in the nuclear cells. They have 0 DNA in the mitochondrial cells. Okay, and so what I just said to you is that there's mitochondrial DNA is something that you may not have heard about before. Another thing, we'll talk more about I talked about the cytoskeletal elements here, the internal structure of the cell. 2 examples of internal structure of the cell that have become particularly important are the centrioles and spindle fibers. Those centrioles and spindle fibers help the cell's internal structure, but in particular, they help during cell division. So the spindle fibers, you can see this right here, called microtubules, and I mentioned microtubules, these little tubes that basically internal structure. During normal operations of the cell, when the cell gets ready to divide, the centrioles then get located in one place and they separate and they get moved. How do they get moved? They get pulled by things like these microtubules and then these actin microfilaments. They get pulled, they get moved from one side, from one area of the cell to another, and they get separated. And then that sets up that process for cell division. So centrioles and spindle fibers are specialized structures that are part of the cytoskeleton. I haven't mentioned a lot of distinctions about the cell membrane. The cell membrane has specialized extensions here. When those specialized extensions are numerous and can be a number of different sizes, those are called cilia. So now, a cilia can also be called a villi, V-I-L-L-I, or a microvilli, little villi. Cilia, villi, and microvilli are the same thing. When we get into this later on this semester, we'll notice the difference. But for right now, what is a cilia, microvilli, or a villi? Here, there's cellular extensions here. So if I were going to draw the cell, imagine I'm drawing my cell, and I don't know if I drew it last week. Sometimes I do. If I'm drawing a phospholipid bilayer and I go like this, and there's just sort of wavy parts right there, I don't think I did. That would be called. These things would be called cilia. There's cell extensions here. They're numerous, and they're small. They're small in relation to the rest of the body. So they're sort of fingers off of the cell. Okay? So that's one thing here. So cilia, villi, or microvilli. Excuse me?
It's like extra tissue.
Not tissues, they're extracellular extensions, extra part of the cell. So these cilia are there are numerous, and there might be hundreds or thousands. And what they do is they wave, like if you're at a concert or something like that and everyone's doing a wave, right? So you stand up and you wave your hands and you sit down, right? That idea, that's what the way cilia work here. They take stuff from one area and they push it over to the other area. If you, for instance, mucus in the respiratory tract, that's an example. I was thinking of other things too. But if you want to get stuff out of your respiratory tract, you capture it in the mucus, so dust or particles or something like that. You get it in the respiratory tract, and then the cilia push it up slowly but surely, and then ultimately you get rid of it through coughing. There's also called flagella, and flagella are unique for a number of ways. Number one, the extension is very, very large, and it causes the cell in which it's found to be able to swim or to move. So this is the only cell in the body that is motile relative to the rest of the body. In other words, it moves in and of itself. I can move across the stage in front of you. You guys can move too. Right? But all of your cells are moving together, whether they want to or not. In this case, the sperm cell is moved by a flagellum. It moves from point A to point B. Ultimately, if there's fertilization, it goes to the egg. It moves from point A to point B through the use of that flagella. So flagella is a very, very large structure here. And if you look at this picture right here, these structures right there are individual cilia. Here, and I'm not going to ask you the structure of the microtubules. I said cilia and microvilli or villi are very similar. Right here, for right now, we're going to say the same thing. If you look at this photomicrograph on the right-hand side, this is an actual small section of the cell. This is a cell membrane here, and it's just going back and forth, right? There's lots and lots of individual extensions of the plasma membrane. All right. Let's see. Jaharlan texted me earlier and said, I want you to go really, really in depth about the cell cycle. Talk about it till 12:30. But I'm not about that. She literally, you want to see my text from her? She said, please talk about it till 12:30. what we're gonna do, we're gonna stop lecture right here. Everything that we talked about today or last week, last Wednesday, is gonna be on the quiz tomorrow. It's fair game. Starting with the cell cycle here, none of this will be on the quiz tomorrow. Okay? This cell cycle stuff we'll talk about after the quiz, so I'm sorry to disappoint you. Too late. So this will be on the, I'll talk about this after lecture tomorrow, after the quiz tomorrow, and it will be on the exam. Okay? So everything up to the cell cycle will be on the quiz. Yes?
Mister, how do you know that?
Gertrude, what?
Dr. Tyson,
Oh.
how do you know that? Didn't you say there's like 300 questions or something, and
Well,
it's all like divided? You're just
so-
gonna have to go back and revisit it.
So what I have done is I've categorized I've grouped the questions into these really small categories, and one of them is called cell cycle, so I just did work that. And by the way, let's just say, so look, so for everyone in this room, right, if I screw up, and I'm absolutely gonna screw up at some point, and if I do, tell me, okay? But if I screw up and I put cell cycle questions on there, then you should say, hey, you told me that cell cycle wasn't on here. So here's what you're gonna do, don't stress out, guess, tell me what number it is, and then you get it, whether you get the point whether you got it right or not, okay? So if I ever mess up, call me, and I'll be able to go back and see your quiz or your exam, okay? Any questions for me?
Sylvia, are you
I'm
recording?
sorry I had to disappoint you. You're not waving to me?
Yeah.
Oh.
Wait, but what does a 9 and 2 arrangement mean?
So the 9 and 2 means that it's the structure of the cilia, and what it has to deal with is how it functions in the context of the actin microfilaments. It also has to do with evolution. It's not something we're going to talk about. So I'm not going to ask you about the 9 and 2 arrangement.
Okay.
Okay?